Thank you very much for the kind introduction. And I am very excited to present our latest technology today. And although I will be talking mostly about spatially resolved proteomics, I actually would like to open the presentation by discussing the complementary technologies, such as spatially resolved genomics and transcriptomics. In the past 2 decades, we observed a tremendous progress in RNA and DNA sequencing techniques that even allow to map single molecules with subcell resolution in tissue samples. In 2020, Nature Methods selected spatially resolved transcriptomics as a method of the year. Despite this chart is only 2 years old, it's already greatly outdated. Only in the past week, there were 3 new methods for spatially resolved transcriptomics reported. And it's really hard to follow the updates. And genetics and transcriptomics launched the precision medicine revolution. These technologies have yielded some breakthrough markers for risk prognosis and the prediction of a drug response and treatment. However, as we get more and more data, we also start to realize that not all genomics and transcriptomics markers demonstrate outstanding ability to predict treatment outcomes in clinical studies. Individual genetic variations, such as a single nucleotide polymorphism, generally account for a modest proportion of phenotypic variability and relatively small increments in risk, less than 1.5-fold. In most cases, this is not enough to justify clinical intervention or changes in treatment. While genomics defines potential gene products, proteomics better reflect the actual phenotype. Recent proteomic characterization of more than 650 cancer cell lines with the previously published genomic and transcriptomic and drug screening data showed that protein expression data correlates significantly better with drug sensitivity or resistance than even mRNA data. In another recent study, it was demonstrated that correlation of single cells, transcriptome and proteome, is less than 50%. Therefore, in the recent years, proteins are gaining more and more attention as both pathological biomarkers and drug targets. One of the major goals of modern medicine is to reveal the correlation between pathological conditions and the proteome perturbations. However, in most of the cases, we actually do not know what specific protein or protein biomarkers are involved in various pathologies. One challenge of proteomics we have to deal with is extreme protein complexity. And this complexity arises from the fact that each gene produces several protein variants far beyond what can be predicted from DNA or RNA sequences. While there are only about 25,000 genes in a human genome, it results in more than 1 million proteoforms. Single cell easily encompasses hundreds of thousands of different proteoforms, reflecting the true chemical diversity of protein molecules, each having a potential to alter cellular structure, function, and interactions. Protein complexity justifies untargeted proteomics as one of the most efficient and well-established methods for biomarker discovery. For example, over 80% of the studies for a new biomarker discovery are solely based on untargeted proteomics technologies, while only a small portion, less than 10%, used exclusively targeted proteomics. And mass spectrometry is the most powerful technique for untargeted proteomics. MS-based proteomics provide an advantage over antibody-based techniques and fluorescent proteins in that it's highly multiplex and requires no a priori knowledge of protein targets. It also retains information about the truncated and post-translationally modified proteoforms. In particular, data-independent acquisition using liquid chromatography, tandem mass spectrometry offers superior depth and throughput of proteomic analysis among all mass spectrometry-based methods. And most proteomic studies are done on a tissue, and tissues are heterogeneous mosaic of cells in different physiological stages and this creates a pressing need to perform spatially resolved proteomics. For example, spatially resolved proteomics is required to elucidate changes in cell biochemistry that occur in early stage of a disease, which might otherwise be unresolvable by conventional bulk-based measurements, as grinding up a whole tissue for the analysis will dilute the contribution of low abundance pathological cells. Currently, there are several different approaches to spatially resolved proteomics. One of them is hydrogel extraction. In this approach, pieces of a hydrogel rehydrated with an enzyme solution are placed onto the surface of a tissue, leading to a specially targeted protein digestion and peptide extraction into the hydrogel. Peptides are then washed from the gel and analyzed by LC-MS mass spectrometry. Another approach is liquid extraction surface analysis, or LISA. A small volume, approximately 500 to 3,000 nanoliters of extraction solvent is placed on a sample surface using a special nozzle. Analytes present in the tissue diffuse into the solvent that is subsequently aspirated over tissue and analyzed by traditional proteomic workflows. LISA can be directly coupled with online LC-MS machine, speeding up the analysis, which is a great advantage of this method. The most common for spatially resolved proteomic approach is based on a laser capture microdissection, LCM. In LCM, a laser is used to perforate the tissue and effectively cut out the target region under a microscope, separating it from adjacent tissue. The separated tissue is then collected using a non-contact approach such as laser-induced forward transfer and process biomass analysis. However, all these approaches have limitations and drawbacks. Hydrogel extraction and LISA have limited spatial resolution, and LCM-based methods require expensive and sophisticated equipment, and also have limited throughput. We wanted to develop a new accessible method for spatially resolved proteomics which can overcome all these limitations, and we decided to take a drastically different approach. We thought, what if we can physically magnify the sample itself so the sample dissection can be done manually with a higher precision without requirements of any special equipment? For physical magnification of biological samples, we utilize hydrogels that can swell enormously in a pure water within minutes or even seconds, as it's shown in real time in this video. The superabsorbing polymers consist of a long chains of atoms that collapse into each other, forming a dense networks. And if we could zoom in to see the molecular structure, it may look something like what you see in this animation. In the collapsed state, the distance between polymer chains with long wide structures are comparable to size of an average biomolecule in our body. When we add water molecules get in between the chains through a process called osmosis and push them apart, making the polymer material to swell about 1,000-fold in volume. Since this polymer chains and distance between them comparable in size to biomolecules, we started thinking, what if we can synthesize the hydrogel polymer within the tissue so densely and so evenly that proteins can get incorporated into a polymer network and allow us to blow up biological sample on a molecular level while preserving proteins' relative location. And we found a simple way to do it in 3 simple steps. First, we treat sample with a chemical anchor that install handles on the proteins, which you can see in this animation as brown structures. These handles will be used to attach protein to polymer chains and eventually help pulling them apart. Next, chemically modified tissue is infused with the monomer building blocks or monomers as we call it. Then we initiate a chemical reaction, polymerization. So monomers self-assemble into long chains, finding their way around between and within biomolecules. Importantly, during polymerization, proteins get tightly attached to a polymer mesh. This will allow eventually pulling proteins apart. When we treat warm tissue polymer composite with a special chemical to remove interaction between biomolecules, so they do not resist to expansion. And now when we add water, that polymer material is going to swell and polymer chains will move apart, as you've seen in the animation before, but now the biomolecules will come along for a ride. Importantly, sample expansion is isotropic on a molecular level, or in other words, relative position of proteins molecules remain the same in the expanded state. Let's see how it looks like for a tissue sample. Here we have a slice of mouse brain tissue expressing a yellow fluorescent protein and visualized using fluorescent microscopy. The slice was embedded into a polymer and treated as I have just described. When it's submerged into pure water, you can see how this brain is expanding before your very eyes. This video is speeded up for about 100 times. Overall expansion process takes about 1 hour or so, and the size of a brain tissue can increase by up to 8 times in linear dimension. And after expansion, you start to see details that were not visible before, like these small dots, which are actually cell bodies of single neurons, but also indicate that proteins retain an expanded state. Expanded samples can be stained with colorimetric dye, such as Kumasi blue. This facilitates efficient visualization of morphological details of expanded brain tissue by naked eye, or other simple imaging setups, for example, smartphone. Imaging of expanded sample allows mapping proteome profile into tissue morphology. Stained samples can be easily dissected by hand with about 100 micron precision using biopsy punch. Dissected pieces of samples are subjected to optimized in-gel digestion protocol to recover peptides for mass spectrometry analysis. Using TIMPO Pro mass spec, we were able to identify more than 4,000 proteins in 3 millimeter punches of expanded brain tissue. And this sample corresponds to about 5 nanoliter of tissue before expansion. This experiment demonstrated that significant amount of peptides can be extracted from expanded tissue, and quality of these peptides was sufficient for mass spec analysis. As a result, we established a straightforward method, which we call proteomics. Proteomics consists of 5 major steps, including tissue expansion, staining, imaging, and microdissection, followed by MS analysis of peptides recovered from expanded samples. The whole process from a fixed tissue to MS analysis takes less than 3 days, and you can see sample preparation for MS analysis does not require any special equipment and can be performed only with the commercially available chemicals and reagents. That makes this technology highly accessible for an average lab. It only involves the steps of a chemical treatment of a sample. Since proteomics involves chemical treatment of an expansion of a tissue, it's important to quantify the efficiency of peptide extraction and validate the qualitative and quantitative reproducibility of peptide and protein identification. First, we decided to measure peptide yield and miscleavage rate as the major parameters for sample preparation quality control. For comparison, we used in-solution digestion and a pressure cycling technology, assisted tissue digestion, or as abbreviated as PCT. Both of these techniques are well established for tissue treatment in MS-based proteomics and often used for PFA-fixed tissue samples. Processing of an adjacent mouse brain tissue section using the selected methods revealed that peptide extraction yield for proteomics was about 1.4 to 1.7-fold higher than that for the solution in digestion and PCT. The total yield of a peptide was about 72 micrograms per milligram of tissue, and proteomics was characterized by a lower number of miscleavages, about 20%, and this miscleavage rate for in-solution and PCT protocols were about 28 and 24% respectively. We believe that the higher efficiency of peptide digestion and extraction achieved with proteomics can be probably explained by molecular decrowding in the expanded state that provides better access of enzymes, molecules to the proteolytic sites on the proteins. Next, the extracted peptides were analyzed using timTOF Pro mass spectrometer in data-dependent acquisition mode. By processing about 200 nanograms of peptides, we identify, on average, more than 30,000 peptides for each method, corresponding to about 4,000 individual proteins on average per sample. The diversity of all identified proteins with three methods had an average overlap of about 66%, and each method uniquely identified less than 7% of a total number of proteins. The overlap for the identified proteins was reasonable and high enough to indicate that proteomics performance was comparable to other methods used for sample processing in MS-based proteomics. Furthermore, all four methods exhibited similar distribution of identified proteins by biomarkers, subcellular localization, and biological function. This indicates that peptide recovery using proteomics is not affected by protein localization or function, and produce protein identification indistinguishable from other methods. Staining of expanded tissue with colorimetric dye facilitates visualization of fine morphological features with a naked eye. This also allows precise targeting of a region of interest by manual microdissection. Therefore, we decided to explore the volume-dependent limit of tissue microsampling using proteomics approach. Using biopsy punches with diameter from 1 to 6 millimeters, we dissected about 8-fold expanded mouse brain tissue. Use size of punches corresponded to lateral resolution from about 120 micron to about 730 micron pre-expansion. We analyzed microdissected sections using pulse DIA mode on timTOF mass spectrometer and identified about 928 proteins for the smallest samples and more than 5,000 proteins for the largest sample. You can see the complete results for the number of identified peptides and proteins plotted against volume of dissected tissue. As expected, the number of identified peptides and proteins increased with tissue volume, reaching a plateau at around 10 nanoliter of tissue. At the same time, PCT-assisted sample preparation, as representative method for processing small samples, enables effective analysis of tissue volume in the range from 0.2 to 1 microliter, which is more than 2 orders of magnitude higher than that for proteomics. Thus, proteomics provides a new strategy for tissue proteomic analysis for sub-nanoliter volume samples, preparation at about 100 microns of lateral resolution. To access the applicability of proteomics to various mammalian tissues, we perform expansion and MS analysis of 3 different mouse tissue types, including brain, liver, and breast cancer. The isotropic expansion is essential for precise mapping of special proteome distribution to pre-expanded tissue morphology. We first quantified the isotropy of hydrogel-based tissue expansion using a non-rigid registration of pre-expansion and post-expansion images. The calculated distortion error were about 8% for the brain, 10% for the liver, and 8% for breast cancer tissue samples, respectively. It means that for about 100 micron dissection, there is less than 10 micron deviation upon expansion. Next, we process about 5 nanoliter volume of each tissue type using DDA-MS and identify more than 2,500 proteins in each sample. Having demonstrated that proteomics enables straightforward proteomic profiling of sub-nanoliter volume of tissue at hundreds micron lateral resolution, we applied this technique to characterize proteomic heterogeneity of mouse brain with and without Alzheimer's disease. We use APP-PS1 mouse model of AD and wild-type mice as a control in 2 age groups. Our primary focus was on the hippocampus as one of the most predominant regions of AD pathology. And the Kumasi stain expanded brain tissue had distinct anatomical landmarks allowing to pinpoint sub-regions of interest by naked eye without the need for any imaging system assistance. From each mouse brain, we dissected multiple subregions with about 30 micron lateral resolution. We selected and dissected primary visual area of a cortex, V1, hippocampal field CA1 and CA3, dentate gyrus, and medial geniculate complex. Microsampling was done manually using a 2 millimeter biopsy punch after tissue expansion, which enabled selected and reproducible microdissection of selected subregions. In total, we prepared 104 hydrogel samples from 12 mice, processed them in parallel using optimized gel digestion protocol. This experiment took less than 5 days, and after MS analysis, we were able to identify about 6,233 proteins in total from all samples. Since the proteomic changes were more pronounced in the old groups, we focused on the old mice for further bioinformatic analysis. In the old groups, the proteomic alternation in AD mouse brain varied among different regions and subregions. At the regional level, there were 73 differentially expressed proteins in the hippocampus, while only 6 in the cortex and 1 in MGC. These results are in agreement with the previously reported findings. Hippocampus is one of the most affected brain regions in AD. Therefore, we further investigated the subregional changes of the proteome in hippocampus. And we found that CA1 had the most substantial alternation with about 198 differentially expressed proteins, followed by CA3 with 19 differentially expressed proteins, while dentate gyrus had no obvious changes, suggesting that changes of AD lesion were not spreading in the entire hippocampus, but in the subregion of hippocampus, mainly CA1 and CA3. We were able to identify individual proteins such as apoprotein E, syntaxin-binding protein 2, as well as proline-rich protein 7 and vesicle-associated membrane protein 1, which were changed in CA1 and CA3 hippocampus subregions, but not in the dentate gyrus subregion. More importantly, ApoE and syntaxin-binding protein were common differentially expressed proteins in the multiple regions, while proline-rich protein and PLAMP1 were only differentially expressed in CA1 and CA3. Altogether, these results demonstrate that the proteomics workflow enables effective investigation of pathological heterogeneity of AD on subregion level in a high-throughput fashion. In all examples above, the expanded samples was stained using Kumasi blue, so we can see it with the naked eye. But fluorescent probes such as antibodies or small dyes. This allows imaging expanded cells. We can increase resolution of optical imaging by that corresponds to a linear expansion factor. Here are some examples of tubulin images acquired before and after expansion using conventional diffraction-limited microscope. In the post-expansion images, we can clearly see nanoscale structures which were not resolvable before expansion. It means that we can perform super-resolution imaging of expanded samples using ordinary microscope. And even linear expansion, for example, for proteomic samples, about 8-fold in linear dimension, then we can achieve about 50 nanometers of spatial resolution using diffraction-limited microscopes. To implement this feature of proteomics, we screened and found a set of small dyes which provide efficient and fast staining of expanded samples. Importantly, these dyes do not interfere with the downstream MS analysis. Here are examples of super-resolution images of different tissues acquired using confocal microscopy, and the tissue samples were expanded using proteomics protocol and then stained with fluorescent dyes for proteins, DNA, and lipids. These features to pinpoint region of interest of proteomic analysis based on super-resolution imaging of cellular and subcellular morphology. Expanded tissue sample is expanded, stained, and imaged. Fluorescent images are analyzed and region of interest is selected. And using targeted microdissection, region of interest is removed and processed for MS analysis. Proteome profile can be then correlated with nanoscale features of the cells in the analyzed region. This is an unprecedented capability of combination of super-resolution imaging and mass spec analysis. And in the end, I would like to say that proteomics is a very versatile technique. Its application can be further extended for multiplex biomolecular detection. Using chemical anchors that react with a nucleic acid and proteins, we can preserve both proteins and mRNA in expanded tissue. Next, mRNA can be analyzed using in situ sequencing, followed by MS analysis of proteins. And we are looking forward to wide adaptation of proteomics techniques for spatially resolved untargeted proteomic analysis of biological tissue and its further multiplexing with other biomolecule species analysis. Development of proteomics technology was done in a close collaboration between Molecular Bioengineering Group and GOMICS Lab, led by Tian Gu at Westlake University. In particular, I would like to acknowledge Ciji, Tin, Shuchan, and Hanbin from my lab, and Judy, Liu, Yaotin, Zhen, Yan from GOMICS, who supported and performed most of the experiments I presented today. This work was also supported with a generous funding from Westlake University Foundation, Natural Science Foundation of China, and Brain and Behavioral Research Foundation. Thank you very much for your attention, and I will be happy to take any of your questions. Thank you.